excluding these variables in combinations to determine the effect of the inclusion or exclusion of these variables on the model. These additional models were then assessed by their area under the curve, or AUC values. An AUC value of 0.5 indicates a model that is no better than if the data inputted was random, indicating poor predictability. An AUC value of 0.7 is considered to be a model with good predictability. We selected the model with the highest AUC value for further analysis. And in this case, it was a model that was created without the bioclimate data set. The habitat suitability map that was generated using the final model. The warmer colors indicate areas of good black rail suitable habitat, with red being the most suitable. The cooler colors indicate areas of less suitable habitat, with blue and gray being the least. Many sites of habitat suitability were identified, in addition to sites of known occupancy. The high-definition version of this map can be used as a tool to find sites that might currently support black rail but have not recently been surveyed, and to identify candidate areas for protection and management. Comparing this map to the estimated species range, the map of predicted suitable habitat significantly distills down the area predicted to be most suitable for black rail. The relative importance and influence of different variables on the model are indicated here in these two tables. The table to the left shows that the land cover vegetation class and the distance to water both had the highest percent contribution and permutation importance on the model. The change in model gain, or effectiveness, is illustrated in the jackknife plot to the right, where different variables were excluded, the green bars, or used exclusively, the blue bars. A long blue bar, or a short green bar, indicates that the variable was very influential on the model. In this case, the land cover vegetation class had the most influence. The model also produced graphs showing which values and categories of each variable were most predictive and associated with black rail presence. I'll go over some of the more important ones in the next few slides. Starting with the most important and influential variable, the black rail response was most influenced by land cover vegetation class. Previous studies suggest that dense water-associated emergent vegetation was important to black rail. As expected for marsh-dwelling species, flooded vegetation, shrubland, and water had the strongest association with black rail presence. The next highest ranking variable was the distance to water. Other studies have shown that black rail tend to stay near, but not necessarily on, open water. This model shows that exact trend with a sharp decline in black rail predictability as the distance to water increases. The third ranking variable was the landform topography classes. The category that this variable was the flat or nearly flat plains, which also supports our hypothesis due to the fact that marshes typically do not have much topography. Previous studies in California have mentioned the potential importance of a gentle land slope to providing variable water depths for the black rail to exploit. The relation between black rail predictability and slope created by the model supports this trend. The highest probability of black rail occurring at flat or nearly flat slope and significantly decreases as slope increases. The effect of anthropogenic disturbance and land conversion on sensitive species is of concern, so we included human population as a variable. There is a noticeable decrease in black rail probability as the relative human population increases. In conclusion, we produced a statistically robust model that identifies areas of known black frequency and identified habitat characteristics consistent with high marsh habitat and also consistent with other habitat association studies. The map generated by the model identifies areas of suitable habitat that could be targets for survey efforts and inform management and restoration. We would like to thank my committee at East Carolina University, the members of the McRae Lab, the ECU Department of Biology, the North Carolina Wildlife Federation Scholarship Program, and the U.S. Fish and Wildlife Service Cooperative Ecosystem Studies Unit. Thank you.
Thank you, Amberly. We have plenty of time for questions. See if anything comes up in the chat here.
Hi, Amberly. This is Amy Schwarzer, Florida Wildlife. And I was wondering, how do you think things like accessibility might bias the model? And for instance, the most recent data from the Everglades shows that it might be a really important, if not the most important area in Florida. But it's one of those things that not only citizens and birders don't get out into, but researchers barely get out into it because of the logistical challenges. So how might that influence? I noticed that the Everglades overall actually did light up, but that core area in the middle. And so what are your thoughts about that?
So this is obviously something to think about when using citizen science data, because of course, citizens are going to report from areas where they want to be. And as you said, the middle of the Everglades is not very physically accessible. But that's the beauty of using this kind of model, because it's those points that the organism is known to be in and then extrapolates it to the rest of the study area. So it looks for places that look like and have the same environmental characteristics as where the organisms have been found, but maybe they haven't been reported there. So yes, you're right, the data being put into it is probably very biased with that, but the model extrapolates to those areas that haven't been, where there might not have been observations, but it still has areas of good habitat. Pardon me.
Yeah, just to clarify for our schedule, there was a typo on the original schedule saying that the second talk starts at 13:30. It actually starts at 13:20. So we do have a couple more minutes if anybody else. Let's see. Nellie, you had a question here.
Sorry about that.
That's okay, Emily. Did you want to finish up saying anything or?
No, did that answer the question successfully, do you think?
I think so. So Nellie asked, can you explain how slope was defined? It seemed to not contribute as much to the model as she would expect.
So the slope data set is from the Esri ArcGIS data set, and it's defined as the difference in elevation from one raster cell to another. So if one raster cell is considered, I don't know, 20 meters above sea level, and then the other is considered like 10 meters below, 10 meters above sea level. So there's that difference in slope. And that was how that data set was created. So that is what the slope is defined as. And yes, it did not necessarily, by the nature of this data, some variables are going to contribute to the model more than others. And yes, the slope was one of the lower ranking contributors to the model.
Thank you.
Time for one or two more. Anybody else?
I'd like to ask another question. Have you gone into the data that has been collected at various states through actual surveys, on the ground surveys, not the eBird type data, to validate the model?
So I did not have access to that sort of data, or I haven't looked into getting that sort of data. And the thing about other survey data is that it might be done differently, or it might look at smaller areas. You know, it might be confined to a certain refuge, or you know, very certain like study area plots, where the eBird data is kind of everywhere. So I wanted a data set that was going to be a lot broader than just, you know, random survey areas from, I don't know, the Atlantic coast. So that's why I used eBird, and it was also easily accessible. You know, that was another thing. So that's why I used the eBird data set, and yes, validating that with like properly, you know, systematically taken data would be a really nice contribution to it, a really nice comparison for other analysis.
I mean, looking at your map of New Jersey, it's spot on with what we've seen with our surveys.
Oh, very good. I'm glad to hear.
Okay, I think we need to move on to the next speaker. If there's any other questions, maybe we could get to them at the end, or Amberly, maybe you could answer those in the chat as well. Our next speaker is Rachel Orban, and she will be sharing her screen here shortly. And she's keeping her video off just to kind of maintain bandwidth, I believe.
Yeah, so I guess that's my cue. I have a video on for just briefly so you can see my face. Hopefully it's coming through. I'll turn it off. And if I am choppy, I guess there's 70 of us on the call right now. So if you do have your video on, it might help to turn it off if I am not coming through very clearly. All right, so thank you for coming to my talk. I'll be talking about cormorant oceanography and characterizing the in situ environment through biologging. And I'd like to start off with acknowledging my co-authors on this. We're all based at Oregon State University, and we're an interdisciplinary team. So there's myself, Adam Peck-Richardson, and Don Lyons are the marine ornithologists of the group. And then we're joined by Dylan Winters and Jim Lurzak, nearshore physical oceanographers. And then Daru Khan Ardig, Greg Wilson, and Tuba Oskin Haller are oceanographic modelers. So why would we even want to use biologging of marine predators to sample the ocean environment? And from the ecological perspective, we often think of marine predators like seabirds as sentinel species. So they're connected to the lower trophic levels and they're visiting biologically relevant regions. So when they go somewhere, they could be sampling this physical environment as well. From the oceanographic perspective, we have a number of different ways that we're sampling the ocean. Oceanographic vessels, satellites, things like buoys, drifters, gliders, and drones. And all of these, combined all of these, we have a number of measurements across the globe, really large data set, but there's still gaps. So the nearshore environment is one of these gaps where it is hard to take high resolution data in the really near shore or estuaries. So using biologging as a tool to sample the in situ oceanographic environment is not a new idea. So the tag on the left of the screen is a tag that was developed for marine mammals, mainly pinnipeds, so things like elephant seals. This tag has been deployed on northern elephant seals, southern elephant seals. So you can see the plot on the right-hand side of the spatial extent of where these tags have been deployed. But they're 400 grams and much too large for pretty much all diving seabirds. And they were deployed first in around 2003, and they're still being used today. So we really need some miniaturization of this type of tagged in order to measure things like water column temperature and especially conductivity, salinity. There has been some work using seabirds, using biologging devices deployed on seabirds to measure the oceanographic variables. So things, tags deployed on penguins to get analogous data to the data set from the seals I was just mentioning. So temperature profiles along foraging penguins, along their foraging tracks. But then there's been also some really creative ways of measuring environmental conditions from seabirds. So birds resting on the surface can be used to measure surface currents. There's been some work to disentangle the influence of both surface currents and wind on at least one species. But you can see on the plot below really good correlation for this data is from razorbills and really high correlation between a modeled surface currents and a bird resting on the surface. And then finally, a really, I think, creative example disentangling wind speed and direction from the stereotypical dynamic soaring paths of shearwaters and albatross. And that's not something that we're planning on doing with cormorants, of course, but it's a really interesting way of pulling environmental data from biologging data. So for the Cormorant Oceanography Project, we are aiming to develop biologging as a low-cost, long-term, and distributed method to measure oceanographic conditions. And to do this for marine birds in particular, we really need tags that are at least less than 40 grams and ideally smaller than that. And then the data from these tags needs to be accessible in near real time, so it can be processed and then accessed and used in models. And then as part of our project in particular, we are using the data in assimilative ocean models. Why cormorants? So there's about 40 species with tropical to polar distributions, and they're found on the shores of all continents. And they're generally larger bodied birds. There are some smaller species in the tropics. And they're pretty active. So they're making 50 to 100 dives per day. And of course, maybe the most important reason of all is that they're using these dynamic nearshore areas that include river mouths and estuaries. And these are places that have really complex and dynamic oceanography. So currently, we're using a tag built by Ornatella. And on this slide, I have the specifications for the tag. And at the top, these are the specifications that the company has basically come with their tag. So they have GSM cell phone communication, a solar panel, GPS, a pressure sensor, 3-axis accelerometer, and magnetometer. And then they've been very generous to work with us, and we've added a conductivity sensor and a temperature sensor. The tags also have really flexible programming, so they can be reprogrammed on the fly through an internet portal. And the sensor data can be collected only during dives. So the pressure and the conductivity and temperature measurements are made when the bird's below the surface. And finally, we've just uploaded new firmware. So now when a bird surfaces, this event triggers a GPS burst and collection of sensor data. And I'll talk about why that's really exciting in a moment. So this is a list of the oceanographic variables that we are aiming to derive from these tags. I've ordered them from easy to hard. And first of all, we're hoping to use these tags to measure benthic soundings, so bathymetry. And this we can use with the depth, the pressure sensor, and then also it's important to have a GPS location so that sounding can be georeferenced. And you'll notice that all of these measurements are linked to that GPS location, so we can georeference any of these variables. The surface currents is something that we're deriving solely from the GPS. And I'll talk about that. I'll talk about all of these in a second. Temperature profiles are reliant on a fast response temperature sensor, wave statistics, the accelerometer and magnetometer. Salinity profiles, we need a conductivity and a temperature sensor. And finally, air-sea temperature contrast is something that we would like to measure. We're pretty confident we can measure the water temperature, but there's some complexity in measuring the air. So this is a schematic that shows you a foraging cormorant, so pulls all these variables together. I have my square coast on the left-hand side, and it is important to that these birds have this really near-shore coastal distribution so that they stay within cell phone range, so they're able to transmit the volume of data that the tags are capable of collecting. And then you can see how when a bird dives, it can measure the temperature and salinity, and then the rest periods in between the dives are really important. So during those times, we can measure surface currents and waves, and hopefully eventually the air-sea temperature contrast. So I won't read this slide aloud to you, but I just wanted to put the following slides in a bit of a perspective in terms of a timeline. I'll be using data from a couple different deployments on a couple different species to illustrate the various variables and how far along we've come in terms of developing those analyses. But initially we used archival tags deployed on Brandt's cormorants in 2013 at the mouth of the Columbia River. And most recently we've been working with pelagic cormorants, and hopefully, or we're planning on working with Socotra cormorants in the next couple of weeks. And finally, our goal is to deploy these tags on a number of different species and hopefully some smaller bodied species. So one question that I think is a really important question to consider is how many birds do we need to tag? And it's something that we think about quite a lot in terms of the ecological context and what questions we're asking when we want to deploy biologging devices. And there's a little bit more complexity when we're considering if we're characterizing the environmental conditions in a region, and you know, how much data do we really need to inform models? We do know that cormorants are showing high spatial fidelity to foraging areas. And even when they're not breeding, we're seeing Brandt's cormorants are returning to the same places to roost. And you know, certainly each oceanographic variable may have a different ideal number of birds. Bathymetry might be more easily characterized. than some more dynamic variables. And we do see as well just changes in activity and location through the year. So the plot on the far right-hand side of your screen shows the full extent of our deployment of Brandt's cormorants. So we tagged birds in the Columbia River, but then as it moved into the winter, we had most of the birds moved into the Salish Sea, but we did have one bird that went all the way south, stopped by the San Francisco Bay Area, and then south to Long Beach, California. So there can be quite a lot of variation in just the movements of the individual birds and where they might be sampling. So benthic soundings depend on both the pressure and the GPS for georeferencing. And of course, not all dives are benthic, but we're using the dive shape to characterize benthic dives. So flat bottom dives are probably to the sea floor, the estuary floor. And certainly birds are not going to provide false bottom, so they can't dive below the sea floor, but we could err too shallow. And you can see some of that. I think you might be able to see by cursor, but you can see some of that on the plot at the bottom here, where there are some birds that dove below the estimated USGS bathymetry. Surface currents. So we've used data from archival tag deployments that were collected at 1 second and 5 second resolution. And this has really helped us sort of hone in on what we might need to collect this type of data from the tags. And thus far, it seems like we really do need high frequency GPS fixes over 8 to 15 second interval in order to get a good surface current estimate. And that's why linking the GPS to the surfacing event of a bird for a GPS burst is such a good solution to this problem, because then we know that the bird's at the surface. We can get our fix when it just comes up from a dive, and then wait for the next dive for the next measurement. So the next measurement are wave statistics, and these are also, we're targeting those surface intervals between dives and using the accelerometer and potentially the magnetometer. We haven't validated this for our free-ranging birds yet, but we've been able to do some tests with a duck decoy in the Hinsdale Wave Research Lab at OSU, and been able to decompose the accelerations to the wave periods that were happening in the wave tank. And we're applying methods that were developed for wave buoys and drifters. And we do expect cormorants to be resting during these periods between dives, and hopefully that will allow the accelerometer data to be reasonably noise free. In this case, the noise is the movement and behavior of the bird. So the next variable is temperature profiles, and this really hinges on a fast response temperature sensor. So we deployed the first tags that had temperature sensors, and also the cell phone connectivity on Middleton Island in the Gulf of Alaska this summer in July and August. And we found that through paired CTD and tag tests, we did note a lag in water temperature measurement. I think it's a couple seconds. And we would definitely like to improve on this lag. And it's because the temperature sensor is quite quick, but it's still embedded in the housing of the tag, and the tag itself carries heat. So the response of that sensor is dampened by its environment, on the tag environment. So we're hoping to make improvements with this component in the near future. But we do think that the temperature profiles that we have from these tags are usable, so which is quite exciting. So the plot here shows a series of dives made by a pelagic cormorant and the temperature gradient that it was encountering while foraging. So the last measurement that I'll talk about is conductivity, and this one really is challenging to develop such a small sensor, to put on tags of this size. And we're really excited to get very promising initial results when we paired the tags with a CTD and dropped it in the water in Oregon this summer. But once we put these tags on birds, we did find that the conductivity measurements were drifting through the duration of the deployment. So there's still work to be done to stabilize the measurements. And we did expect that drift might happen with this type of sensor. So this is another thing that we're hoping to improve. So what's next? Reduce the response time of the temperature sensor, probably by making it more external to the tag. We're hoping to try some alternative materials for the conductivity sensor to reduce the drift. The sensors that we're using now are made out of gold. Would like to continually miniaturize the tag and refine our attachment methods and gain a better understanding of how many birds we might need to tag. We are working to automate the process of taking the tagging data from the bird, pulling it into the animal telemetry network, and in that process, standardizing the data and allowing it to be used. So we're working on building that automated pipeline. And we're hoping to develop collaborations to deploy tags on new species and continue our assimilative ocean modeling efforts. All right. I guess I can take any questions now.
So Rachel, you've run out of time and I didn't want to interrupt you there, but maybe if there's time at the end, or people can type things into the chat and you can answer them directly that way.
Yes, of course. Or I can, I'm also happy to field questions by email if anybody's interested in something specific.
That sounds great. Thank you. Very interesting. Okay, the next speaker that we have is actually a student, Michaela Call.
Okay, can you hear me?
Yes.
Awesome. Okay, so good afternoon, everyone. Today, I'm going to present some of our findings and lessons learned from using camera technologies to remotely monitor American oystercatcher reproductive success on a Virginia barrier island. Okay, it's not advancing. Hold on, let me reshare my screen. Okay, now it's working. So our study was located on Wetumpka Island, which is part of the Virginia Barrier Island system off the coast of the Delmarva Peninsula. It's a unique wilderness area that is an important breeding, migration, and wintering location for over 100,000 shorebirds on the Atlantic Flyway. Wetumpka Island is protected and managed by the U.S. Fish and Wildlife Service, as well as the Nature Conservancy as part of the Virginia Coast Reserve. It's a remote location, only accessible by boat, so the impact from human recreationists on the island's breeding shorebirds is low relative to other locations on the U.S. Atlantic coast. However, its location also results in longer intervals during reproductive success monitoring. It's a hotspot for breeding American oystercatchers in Virginia, and it is arguably the most important breeding site for oystercatchers on the Atlantic coast because it hosts around or over 100 pairs nesting on the island annually. This graph shows annual productivity defined as the number of chicks fledged per pair and the number of pairs monitored from 2002 to 2018 on Wetumpka Island. Productivity is on the left axis and is depicted by the blue bars, while the number of pairs monitored is on the right axis and depicted by the red line. So you can see that there's been an increasing trend in the number of oystercatcher pairs that are breeding on the island. Productivity on the island is often greater than the average productivity for the entire Virginia Barrier Island system, which most recently has been reported as 0.61 fledged chicks per pair. It's also high relative to other locations on the Atlantic coast. For example, best available productivity estimates in the species account for oystercatchers is 0.45 fledged chicks per pair in Massachusetts and Maryland, and 0.32 fledged chicks per pair in North Carolina. From 2002 to 2018, productivity on Wetumpka averaged 0.73 fledged chicks per pair. Productivity declined around 2008 and almost complete failure occurred in 2009 due to a fox that was on the island. Productivity increased again in 2010 after that fox was removed. Productivity has declined again in recent years, averaging 0.25 fledged chicks per pair in 2016 to 2018. This has occurred despite continued predator removal on the island, triggering a need to reevaluate the threats to oystercatcher productivity on Wetumpka Island. Our study was part of a National Fish and Wildlife Foundation funded demonstration project, which was conducted at 3 sites along the Atlantic coast, Wetumpka Island being one of those sites. We had several objectives for our study on Wetumpka. We used camera technologies to better understand threats to productivity by identifying causes of nest failure, characterizing the predator community, and quantifying the activity of all non-shorebird species at nest sites and along the marsh edge. We evaluated the accuracy of nest fate assignment from observations gathered in the field in order to understand the accuracy of hatch estimates and the importance of different threats. We also assessed the ability to use cameras for remote monitoring of nest and brood survival. And finally, we used cameras to improve our understanding of visitor use during the breeding season and how it may be impacting shorebird reproductive success. We used 3 camera setups in our study. The first was our nest cameras. We placed 27 cameras on active oystercatcher nests once incubation began. They were set to capture 3 photos per motion trigger with a 5-second reset speed, both day and night. To monitor broods once they had left the nest, we placed 11 cameras at known brood rearing sites. We identified these sites based on field observations of nest locations and oystercatcher territories, as well as knowledge of brood rearing locations from previous breeding seasons. These cameras were also set to capture 3 photos per trigger, both day and night. Finally, we deployed 7 cameras 40 meters apart along a transect that ran parallel to the marsh beach interface. These cameras were placed 50 to 75 meters away from the marsh edge, facing the marsh, so that they provided a wide angle view of the marsh edge. And these were time-lapse cameras, and they took 1 photo per second and ran approximately from 5 a.m. to 9 p.m. Weekly checks for nest and brood monitoring occurred from April 1st to August 31st. We searched all suitable habitat for nests and then monitored those nests until hatch or fail. We defined a nest as successfully hatching if at least 1 egg hatched. Nests were considered failed following abandonment or complete clutch failure. Following a successful hatch, broods were monitored until chicks either died or were considered fledged at 35 days after hatching. When possible, the cause of nest or brood failure was identified using field gathered evidence such as predator scat, tracks, and eggshell fragments. To assess threats to nest success, we extracted predation and disturbance events from each nest camera. We defined predation as any event that resulted in partial or complete clutch loss, while disturbance was any event that resulted in the incubating adult leaving the nest. We recorded all causes of these events, including non-predator species, humans, overwash, and in 1 case, a balloon. This figure shows the causes of these events and their encounter rates defined as the number of events by each cause per 100 camera days. The species marked with a black star are known shorebird predators listed in the Guidance and Best Practices Report for Predation Management on the Atlantic Flyway. I marked herring gull with a red star because it was the source of the only nest predation event captured on camera. Other American oystercatchers intruding on the nest site were the most frequent cause of disturbance, followed by white-tailed deer, grackles, and human recreationists. We also recorded all non-shorebird species observed on the transect cameras in order to quantify the activity of both predator and non-shorebird species at multiple brood rearing sites and along a stretch of the marsh edge. We did not observe any chick predation events on the brood or transect cameras, but we were able to use observations to characterize the predator community. So we were surprised to find that we did not observe any mammalian predators along the marsh edge, and we only observed mammals a few times on the nest cameras. This was likely an indication of the success of the predator removal efforts that occur on the island. Diamondback terrapins were the most frequently encountered species on the transect cameras, likely because of their location along the marsh edge. In a few instances, adult American oyster catchers with a brood interacted with these diamondback terrapins aggressively, suggesting that they perceive diamondback terrapins as a disturbance or threat, despite terrapins not typically being considered a shorebird predator. Grackles were the second most frequently encountered species, followed by ghost crabs. It was difficult to see those ghost crabs when they were located farther from the camera, so it's likely that ghost crabs were more active at the marsh edge than indicated by our calculated encounter rate. Here's a photo of one of those aggressive interactions between an adult oyster catcher with two chicks that are located out on the left of the adult and a diamondback terrapin that was passing by the brood. Overall, we found that field monitoring underestimated hatch success and that camera monitoring improved estimates of hatch success by assigning nests with unknown hatch fates as either hatched or failed. We were able to identify 2 nests with unknown fates from the field as successfully hatched using evidence from our cameras. But why did field monitoring underestimate hatch success? It was likely because field monitoring often relies on the presence of signs around the nest site to indicate hatching or failure, such as tracks or eggshell fragments. These signs are ephemeral and are quickly removed by wind and rain, and at a site such as Metomkin, where monitoring intervals may be 7 days or more, identifying nest fate can be difficult because signs may not be present. Signs may also be obscured by or become confusing due to multiple predators visiting the nest site after the nest had failed. And so here's an example of what I mean by that when I say that signs can become confusing after the nest fails. This is a photo of a herring gull holding an egg in its bill. This is from the one nest that failed on the nest cameras. This nest failure event occurred on July 25th. And then the next day, a grackle was hanging out around the nest and showed up on the camera multiple times. So if a nest check occurred on the 26th or the 27th of July, then likely the cause of failure would have been assigned to grackles because of tracks left in the sand. In the case of this nest check, though, the nest wasn't checked until 6 days after the depredation event on the 31st. Notes from the nest check identified the cause of failure as a ghost crab due to a ghost crab hole that was located in the scrape. The nest camera then showed that this was not correct, demonstrating how cameras can improve the accuracy of both hatch success assignments, but also provide insight into the degree of threat from different predator species. So when assessing the ability to use cameras for remote monitoring, we're also interested in the effect of cameras on nest survival to ensure that cameras are not leading to decreased hatch success. None of the nests in this study were abandoned following camera deployment. We monitored each nest immediately after deploying the camera to make sure that adults returned to the nest, and if the incubating adult didn't return within what we considered an appropriate amount of time, we pulled the camera. Additionally, only one camera monitored nest was depredated, suggesting that the cameras did not seem to attract predators given the predator community that's present on the island. Hatch success, defined as the number of the total number of hatch nests out of the total number of nests, was not lower for camera monitored nests. We intend to model and compare the daily survival rates of camera monitored and non-camera nests to ensure that those daily survival rates are not different between the 2 subsets of nests. We are also interested in assessing the potential to use cameras to remotely monitor brood survival. Hatch success for the subset of nests monitored on Metomkin Island was 82% based on field observations and 91% based on camera observations. So despite low productivity, hatch success does not appear to have declined. This suggests that some unrealized factor may be impacting productivity during the brood rearing stage instead. However, identifying the cause of chick mortality can be difficult. Oystercatcher chicks are highly mobile and cryptically colored, which makes them challenging to locate once they have left the nest site. We were able to observe those broods frequently on the brood and transect cameras, though, from near hatching through post-fledging. We found that it may be possible to monitor brood survival using cameras if we know brood rearing territories. Broods may move in and out of the camera view, but if they're visible at least once per day, we may be able to model survival using a model that allows for imperfect detection. So in addition to monitoring shorebird reproductive success, we were also able to learn more about visitor use of the island. Wetumpka Island is open to visitor use year-round, though visitors are restricted to the intertidal area during the breeding season. Pets are not allowed on Wetumpka Island, though it's clear that visitors do not always abide by this rule. Cameras provided managers with further information on visitor access points, use of more remote areas of the island, and recreational activities, as well as the presence of dogs and the overall threat to breeding shorebirds. In these photos, you can see that visitors pass through shorebird breeding territories, sometimes passing rather closely to a nest, which I have circled the nests in these photos with a red circle. Overall, this information can be useful to improve management of visitors on the island in order to reduce the human threat to breeding shorebirds, such as providing information on where symbolic fencing should be placed. We learned several lessons from this study that I would like to share with you. First, we found that cameras were beneficial for monitoring remote locations, particularly at a site that is more challenging to access and consequently has longer monitoring intervals. The cameras may highlight potentially unexpected causes of disturbance and nest failure, providing the opportunity to adapt and improve management efforts. Cameras also improved accuracy of reproductive success estimates by reducing the number of unknown nest fates. However, we found that we would not be able to estimate nest success based on the total number of eggs hatched, as it was often difficult to see the number of eggs in the nest using the cameras alone. We found that cameras, particularly those located along the marsh edge, were beneficial for characterizing the predator community and may provide important data for locations that are unsure about predation threat. We were not successful in capturing instances of chick predation on the brood and transect cameras. This may have been due to chick predation occurring outside of the camera range. We suggest combining camera monitoring with telemetry to improve understanding of causes of chick death, as telemetry may allow for recovery of some of the chick remains. Important drawbacks to note about camera monitoring were that it produced a large amount of data, which may take hundreds of hours to review. For example, it took 315 hours to review our transect cameras, which captured 322 camera days of time-lapse footage with 1 second intervals. We found that our cameras improved hatch success estimates and did provide insight to Wetumpka's predator community, but managers should consider trade-offs between access to such detailed data and also the effort that it's going to take to review that footage. We also found that the cameras may have attracted visitors, potentially drawing them into the nesting area and near nests when they would not have approached the area otherwise. And so we encourage that managers considering camera monitoring keep these drawbacks in mind when designing their methods in order to ensure that they're not threatening the nests and broods. So I'd like to thank my co-authors and partners with the Nature Conservancy, Virginia Department of Wildlife Resources, U.S. Fish and Wildlife Service, and Virginia Tech Shorebird Program. Funding for this project was provided by the National Fish and Wildlife Foundation. And if there are any questions, I'd be happy to answer them.
Thank you, Michaela. Erica had a question. Did you have any disturbance of your cameras by people or anything else?
So there were photos of people taking photos of the cameras. So they were getting very close to the cameras. And so that's definitely people being attracted to them. I don't think that there were any instances when they actually disturbed the camera themselves, but I feel like that's definitely a possibility, especially if like the photos of people taking photos of the cameras indicate to me that they're very interested in the cameras. So, yeah.
Were they locked and secured to where they were posted, or?
Yeah, so they were attached to like steel U-posts that had been pounded into the sand, and they were attached to that.
Okay, let's see, another question. What cameras did you use and what were the causes of the camera failure?
So we used Let me see, I can actually reshare. I have some more information on the cameras. So we used a combination of cameras. So the Nest cameras were all from Blaze Video, as well as the Brood cameras. The Transect cameras were Plot Watcher 6 Pro, which I don't think are made anymore, and Brino cameras. And a lot of the failure was because they were on a combination of solar power and battery. And so we found that the solar power was not working as expected in some cases.
Let's see. How often, I might have missed this, but how often were you kind of attending to the cameras? Was it every couple days or a week?
Yeah, so the monitoring intervals on Metomkin can be tricky because, like I said, it's only accessible by boat, and the tides on the eastern shore of Virginia can be really difficult. So they were checking on the cameras when possible, and so that, you know, those monitoring intervals could be anywhere from, they were usually around a week, but they could be a little bit longer and sometimes a little bit shorter.
And is there any way you could have scaled down? I'm just thinking of somebody with limited resources, scaling back the number of cameras and still get kind of the information you want and need.
I think you could. I think the amount of cameras we put out, part of that had to do with the fact that there's so many pairs breeding on this island. And so, you know, that's why we had 27 out. Honestly, we could have had more. But again, the problem with cameras is that you can get more detailed data, but that is resulting in a lot more time of going through the photos. And that can be a really tedious process. And we did try using some technology that would be able to identify like motion on the time-lapse photos. And it wasn't beneficial because it was just picking up on every ghost crab, every sanderling, every beach grass. So like it was a lot of effort. So, but I think you could scale it down. That's going to be part of the balance that managers have to find.
Well, thank you. That was great. We have to move on. So our next speaker is also a student, Logan Klein. You can go ahead and start sharing your slides.
One second. Okay, hopefully everybody's seeing my slides. All right, so hi everybody. My name is Logan Klein and I'm a grad student at the University of Maine. I'm going to talk to you today about a component of a larger research project that's being run to increase data literacy in wildlife survey technologies. Specifically, I'm going to talk about a few interdisciplinary projects being conducted at UMaine that address the question, can unoccupied aerial vehicles and novel artificial intelligence techniques improve accuracy and efficiency of nesting waterbird monitoring? So coastal waterbirds such as herring gulls, double-crested cormorants, and great blue herons are highly sensitive to shifts in ecosystem conditions, which makes their individual and colonial success potential indicators for marine and coastal environments. For this project, we're developing methodologies to assist biologists who monitor the status of seabird and wading bird populations off the coast of Maine. There are inherent challenges in monitoring nesting birds, however, that we want to address. So I probably don't need to tell this group that monitoring bird populations is characteristically difficult. In ground-based counts, observers are typically on foot and can count seabirds at close range, enabling them to locate smaller or more cryptic species, as well as distinguish between very similar species. However, these surveys are prone to visibility errors in which a bird or nest that is present is not detected. Furthermore, ground counts can be a source of disturbance to those birds since the observer may walk directly through sensitive habitat. Finally, many locales with nesting birds are inaccessible or dangerous to survey on foot, leaving them unsurveyed or only partially surveyed. Aerial counts function as an alternative methodology to mitigate the risk of ground surveys. Aerial counts work well when you have large subjects like elephants or blue whales, or subjects with bright colors that are highly visible and relatively easy to see and count. However, counts are still subjected to visibility biases as animal group size, behavior, coloring of individuals, and obstruction by vegetation can affect accuracy. A solution to these limitations has been to implement plane-based imagery in which a camera affixed to the plane takes snapshots that can be compiled into a mosaic for post-flight counting. The high altitude at which the survey plane is flown, often above 300 meters, limits the resolution of images captured from plane-based surveys depending on camera specs. Additionally, post-processing imagery to obtain accurate counts can require substantial time. Unoccupied aerial vehicles, or UAVs, carrying cameras have been increasingly used in studying animals ranging from gulls to even crocodiles. UAVs, in comparison with plane and ground surveys, have numerous benefits. They're lightweight and portable, enabling researchers to transport them relatively easily to study sites. They also have automated, programmable flight capabilities. Perhaps most importantly, UAVs provide high-resolution imagery when compared to planes. The resulting imagery provides a snapshot of populations at particular moments in time and allows biologists to revisit snapshots later for manual processing. A variety of camera types exist that can be used to improve information capture for the question of interest. Important limitations to UAVs must be considered as well. Few standardized protocols exist that outline procedures for approaching birds with UAVs or achieving image resolution high enough to reliably identify birds to species and behavior. The sheer amount of manual processing needed to count seabirds from imagery can also be prohibitive. Some scientists have postulated that manually counting birds in UAV imagery actually offsets the benefits of using UAVs in the first place. Various automated tools have been created that can process imagery and create counts without manual input, but a streamlined, user-friendly tool for automated processing does not yet exist on a scale that can be readily employed by biologists and citizen scientists. The lack of a vetted, efficient automated process is a major research gap that we hope to address with our research. So we are collaborating with a suite of specialists, including state and federal biologists, remote sensing technicians, GIS scientists, computer scientists, and undergraduate and graduate students to meet our overall goal of developing efficient automated processes for obtaining accurate estimates of colonial nesting bird abundance using plane and UAV-based imagery as well as ground counts. So we have 3 objectives to guide us in reaching this goal. First, we want to compare the accuracy and efficiency of manually deriving bird abundance, species, and behaviors from historical and recent plane-based imagery, UAV imagery, and ground surveys, and identify sources of error in these estimates. Through a partnership in our computer science department, we are also developing and evaluating automated processes using AI to derive bird abundance, species, and behaviors from historical and recent plane and UAV-based imagery and identify sources of error in these processes. And finally, we want to create an open source platform for state and federal biologists, as well as citizen scientists, to upload their own imagery and take advantage of our artificial intelligence algorithms to identify birds. Although our framework is focused on deriving counts of nesting birds, we anticipate that there will be much more broad applications of our approach and tool sets. So we were fortunate to come into a monitoring program for coastal nesting seabirds that has been conducted over 25 years and has a large data set of plane-based imagery and bird counts developed from those images, as well as a subset of the same locations counted with ground surveys. In order to evaluate the efficiency of incorporating UAVs into monitoring and explore AI processes, we need to assess population trends derived from plane and UAV-based imagery and investigate errors in methodology. The U.S. Fish and Wildlife Service flew aerial surveys to capture imagery of roughly 270 of Maine's coastal islands in 2008, 2013, and 2019. Archival ground counts exist for these years to compare manual imagery counts to. I'll touch on the ground survey protocol in a few slides, but major shout out again to the Fish and Wildlife Service and our other partners for providing this imagery and archival data sets. For the 2008 and 2013 data sets, biologists reviewed the imagery for signs of nesting gulls and cormorants and put a point on each nesting bird they could identify. Part of our work has included processing the 2019 plane-based survey that collected imagery with slight modifications to quantify interpretation error and discriminate behaviors in addition to nesting status. We are marking every bird that we can identify to species and behavior. This means marking all nesting, roosting, and flying birds we see in order to grow a training set for automated processing with artificial intelligence algorithms, as well as examine errors that may exist in our manual species and behavior identification. We're also conducting UAV surveys with the objective of obtaining high-resolution imagery of multiple species of colonial waterbirds. This past summer, between May and July, we conducted 42 UAV flights over an inland great blue heron rookery and 6 coastal islands containing nesting herring gulls, great blackback gulls, common eiders, and double-crested cormorants. We flew a baseline flight at 60 meters above the colonies, then lowered flights to the first signs of disturbance or a minimum of 15 meters. My colleague Meredith Lewis will provide more information about those surveys and bird responses to the UAV in her upcoming presentations, so stay tuned for more details on that. We will be manually interpreting this UAV collected imagery in the same manner as previously described for the plane-based imagery, so that we can compare manual and automated counts with ground counts. Finally, we conducted ground surveys immediately following our UAV surveys to account for visibility and observer errors in our UAV image interpretation. We followed protocol established by Fish and Wildlife Service and Maine Department of Inland Fisheries and Wildlife, in which observers walk wandering transects through sites while counting and identifying birds to species. We also took coordinates of confirmed nests and nesting birds, as we will compare these locations with our manually and automatically interpreted UAV imagery. Unfortunately, we weren't able to and will not be able to conduct ground surveys at all sites out of respect for the birds. Ground counts create a lot of disturbance, and developing methods to reduce that disturbance is a key justification for our research. So now that I've given you context for the data sets we're working with, I want to showcase some of the collaborative projects that we have developed. Given the rich data set we have available, there are exciting opportunities to harness the knowledge of interdisciplinary teams of students to address our focal questions and develop user tools. For example, we spend nearly 840 hours reviewing imagery and clicking on every bird we see. That is 35 straight days. Manual processing can also be mentally and even physically exhausting. It's simply an inefficient process that we want to improve by bringing computer science and biology together. So we're working with Alex Ravello and Timothy Bruce, two Masters of Science students in Computer Science Department at UMaine, and Dr. Roy Turner of Maine SAIL to create a streamlined, automated process that we can use to interpret imagery and identify birds. Ultimately, this work will be incorporated to a user interface that I'll describe in the next section. So we're developing this automated process with deep learning. In deep learning, algorithms are written with multiple processing layers that learn representations of data with levels of abstraction. In this image here, for instance, an algorithm might use multiple combinations of imagery bands or channels to pick out features of a cormorant nest that make it a nest. These deep learning networks need a large data set to train on, which is what we're providing by manually reviewing our plane and UAV imagery. We have multiple objectives in using a deep learning process to automatically identify and count birds. First, we'd like to reduce the processing time and manual labor it takes to interpret survey imagery. More than that, we want to reduce visibility bias and misidentification, as well as be able to quantify that bias and understand how to reduce it through improved algorithms. The trick here is to provide enough information for the computer to reliably identify a target, or a bird in this case, which means lots of images and context that are representative of the focal object. There are two important components that the network needs to include. The first component is image segmentation. This means we need the program to search through large image files and segment out pixels that contain birds. With those segments, the second component will need to classify the bird by species and behavior. The algorithm currently being implemented by the AI team is known as MaskRCNN, or a region-based convolutional neural network. This algorithm creates regions of interest around objects in an image and creates boundaries on them, as you can see here in the top row of images, then segments them from larger imagery and generates masks. The mask component of the algorithm generates the second row of images here, and these masks are used to accurately discriminate among the species and their behaviors. The goal of the MaskRCNN is to generate identifications and report confidence of that classification. For instance, here we have a mock interpretation of output that predicts the presence of 3 great blackback gulls and 1 herring gull with relatively high accuracy. When the algorithm is ready for us to train it and process our imagery, we will compare its predictions against our manual classifications and ground counts when possible. Through another collaboration with the computer science graduate student, we are attempting to look at uncertainty in behavioral classification by incorporating a depth map as a 4th channel in our algorithm. Depth maps are generated by matching overlapping points in raw, unrectified images from planes or UAVs. We programmed our UAV flights with overlap to minimize edge distortion and enhance point matching when images are mosaic together, and we can use this overlap to create depth. Our hypothesis is that these depth maps may capture subtle changes between nesting birds and substrate or vegetation that our eyes can't see, and incorporating them may improve the accuracy of identifying birds by behavior. To our knowledge, this might be the first time depth maps have been incorporated in automated identification of bird behavior in images. So we will be assessing any increase or decrease in accuracy of the classification process in both species and behavior. The second project I want to highlight is a collaboration between computer science undergraduates, biologists, and graduate students in generating data visualization techniques and an open source application that will connect users to those techniques, as well as our artificial intelligence algorithms. Our goal is to facilitate data interpretation by exploring ways to visualize the data sets and to integrate those visualizations into a user tool that enables non-computer scientists to apply our AI processes to interpret their own imagery. So we collaborated with a group of undergraduate computer scientists at UMaine who were interested in helping us visualize the count data we currently have, as well as count data generated by our AI algorithms down the road. The students generated maps like the one you see here that display manually counted birds by species and behavior on an island in our study area. These will be further adapted to include information captured by our UAV flights this summer and the upcoming summer. The team also provided graphics generated for observing user error. This Marimekko chart here shows the differences in identifications of 3 different observers for an island count. Ideally, these can be edited to display the differences in identifications between a manual count, an AI count, and ground count surveys, and can be used in determining observer error. The graphics generated by the data visualization team will be incorporated into a larger open source application, which we're calling BirdSpotter, that we are developing in collaboration with the team of another group of computer science students who are using this project for their senior capstone project. This application will serve as an interface to connect biologists and citizen scientists with the AI methods we are developing, as well as data visualization techniques. We are in the early stages of this work, but we have had several productive conversations with the team that exemplify the power of connecting biologists with computer scientists to use their diverse knowledge and skill sets to address complex ecological questions. The results of this research will have implications for our study populations and beyond. By documenting seabird and wading bird species across a large volume of sites, we will increase understanding of their presence in the Gulf of Maine. This will provide our partners, such as the Fish and Wildlife Service and Maine Department of Inland Fisheries and Wildlife, with valuable information that can help them prioritize species of concern in their management practices. Data collection on sensitive species, such as the double-crested cormorant, can help us map trends of their population decline and better prepare organizations for protecting them from future loss. There is a huge amount of data gained from aerial surveys with imagery. However, processing is currently a notable limitation. Demonstrating that automated processing of these remote image collection approaches can improve monitoring efficiency and accuracy is an important step toward incorporating use of these technologies in wildlife surveys. Additionally, developing the tools and user interfaces that can be applied by biologists without computer science skill sets and backgrounds will address a current need for an accessible, open source, accurate program for classification of species and behavior. Finally, we are fostering collaborative education opportunities and research experience for young scientists, as well as building bridges between institutes of interdisciplinary knowledge to work together to solve complex ecological questions with our combined knowledge. So with that, I wanted to thank everybody on this list, from the Fish and Wildlife Service to Maine Department of Inland Fisheries and Wildlife, to all the landowners that let us access their lots this summer, as well as my academic partner in crime, Meredith Lewis, who again, you guys should stick around and listen to her talk, which is up next. So thank you.
Thank you, Logan. It's kind of mind-blowing thinking about all that work that you guys are putting into that. We have a couple minutes for questions. Let's see. Very cool talk. Ann McKellar says, is the user interface going to be something that a biologist with their own aerial imagery used to perform automated counting? And when do you anticipate it will be available for use?
So yes, that is the goal. Our idea is that, you know, a scientist in Montana, for example, can go out and take pictures of ducks and then upload all those aerial images into this program, essentially. Have the program do some automatic interpretation, spit out some results. As for when that will be done, technically the computer science team that's working on it is supposed to be quote unquote finished at the end of next semester. However, we anticipate probably needing to do some cleaning of that before it's necessarily available for use.
Okay, is that something that when you do input the image there, it kind of trains itself on it? And will there be any interaction between the user to say, well, that doesn't seem right, you know, go back and recalculate that, or here's where there was an error?
Yeah, so we'll probably incorporate images beforehand, before releasing it, of like other species, for example, to do some training, but there's definitely the opportunity to keep on training the algorithm as we move on. And then as for the second part of your question, yes, there will be an interactive part where you can look at the data that's output. And hopefully there'll be a little image that shows what the AI is calling like a common eider, for example. So you could look at it and say, well, no, that's actually a herring gull or something like that.
Another question we have from Kevin Rogers. Do you think the tools you are developing would be able to handle thermal imaging for species that are cryptic from above? And have you been able to mosaic images where birds are not staying still? In other words, foraging instead of incubating.
Right. So I'll answer the second part first. We have definitely been focusing on birds that are incubating so far. However, foraging birds, depending on the height that you're getting the image from, might not be as distorted as you might expect, because if they're only moving a small amount, there's probably not going to be enough overlap, or excuse me, there's probably not going to be too much disagreement between where they are spatially. We have had issues, however, of birds that are flying. When you're taking images of them, it gets kind of warped. So it really kind of depends on the spatial resolution that you're taking the images at. And I guess the purpose of what you're taking images of and why you're doing it. So there's that. We haven't integrated thermal yet. However, thermal imagery is pretty much treated as a separate band. So you're looking at red, green, blue. We're throwing in another band, which is depth. So you could integrate thermal imagery into the training algorithm as well. One thing that we're really interested in looking at is using thermal to detect eiders, which we'll see if that ever happens, but we're really hoping that we can do that, and then we'll incorporate that as well.
Great, thank you, Logan. I think we're out of time for questions, so we're going to move on to another student, who Logan had mentioned, Meredith Lewis. Also, her work relates to what Logan just spoke about.
All right, thank you, Danielle. Can everybody see my screen okay and hear me all right?
Yes.
Awesome. Well, thank you all for sticking around. My name is Meredith Lewis. I am a graduate student at the University of Maine along with Logan, co-advised by Daniel Hayes and Cindy Lofton. And I am the other student, I guess you could say, who's working on this comprehensive project to evaluate the use of unoccupied aerial vehicles or drones to survey colonial water birds, as well as integrating the AI process. And with that in mind, this is definitely like a huge team effort that integrates not just our partners, but also the work of biologists and computer scientists. But today, for the purposes of what I'm going to talk about, I'm going to talk a lot about our preliminary observations from this past field season, that where we did manage to fortunately get out and do some test flights and develop some more protocols that we'll hopefully be able to implement more in depth next season. So I'm going to start by talking a little bit about all of the birds that we know and love, and I don't think I need to sell to you how cool they all are, which is one of the reasons that I'm particularly interested in them. But that we're also interested in them not just because, you know, they're cool and they nest together on islands, which lends them well to aerial image surveys, but they're also sentinels of changes in the marine environment. They're sensitive to changes in food availability as well as pollutants, which makes which sort of emphasizes the importance of monitoring these populations consistently and long term. And some of the ways that we monitor these populations, as Logan mentioned, include ground counts and plane-based imagery, which, depending on your goal, can be either really useful or can also exhibit a lot of disturbance. So in the case of ground counts, for instance, sometimes you don't have, depending on if you're on a cliff or anything like that, some sites may be completely inaccessible to biologists for the purposes of ground counts, which might restrict you to plane-based imagery, which essentially captures a snapshot in time. And then ground counts too also can cause a lot of disturbance. And in fact, some species here in the Gulf of Maine, we don't even walk through the colonies at all. So if you're not walking through colonies to get that information on how many eggs you have in a nest or how many birds are actually nesting there, you might be missing some critical information that is important to the management of these species as a whole. Which brings us to sort of the middle ground we like to say as unoccupied aerial vehicles, which have garnered a lot of attention in recent years. And they are in fact being more readily used by biologists to survey wildlife, particularly colonial birds, such as terns and even gulls. And given that they are becoming a lot more accessible with respect to costs and their portability, they do show a lot of promise. And given that they are advancing so rapidly, we don't necessarily have a complete picture on just one, whether or not certain species are more affected or more susceptible to these kinds of surveys with respect to disturbance than others, but also we don't necessarily have a complete picture of the context in which these types of technologies are best implemented. And we also are looking to gather more data as to really push whether or not we can get different sorts of information other than just an aerial image from one survey event for these types of surveys. So all of these are sort of the inspirations of what I'm going to talk about today. So I'm going to talk a lot about like the biological perspectives and sort of the field biology perspective. So some of the goals and objectives from this past season were to use UAVs to gather some population metrics of the colonial birds, namely abundance of nesting birds, which is generally, I think, something that's of interest to most biologists, is to sort of estimate the size of the nesting colony. But we're also looking through repeat visits and repeated surveys gathering this imagery to look at productivity estimates for certain species. We also are looking to evaluate species-specific behavioral responses to these technologies by capturing the responses of a lot of different species. And then we're also revisiting colonies throughout the breeding season to see how these changes respond based off of the changes that these birds are going through throughout the breeding season, and whether or not birds that are just roosting towards the end of the breeding season might have differences in their response. We're also looking at UAV performance in various habitat types. We're fortunate in Maine that, fortunate or unfortunate in Maine, that we have a lot of islands that are either covered in brush or have various topographic challenges that we are looking to address and prevent challenges to using UAVs, but we're hoping to have a comprehensive set of recommendations, not just for automated interpretations, but also for implementing these technologies, depending on the situation that these birds are in. So I'll go over a little bit of our methods that we use this field season, starting with just a general overview of the methods from field survey all the way to image interpretation. And then I'll talk a little bit more in depth about the behavioral surveys that we started doing, as well as some of the ways we hope to eventually estimate productivity for select species. So we were fortunate enough this summer, in spite of all of the stuff going on in the world, to visit 6 coastal islands and 1 rookery. We did visit more rookeries throughout this season, but one of the challenges of working with these birds is sometimes colonies are not necessarily active year to year. But we did manage to capture imagery from 43 UAV flights from early June to mid-July. And here's some examples of just some of the range of species that we were able to capture, including great blue herons and double-crested cormorants, and some of the islands we were able to visit too. So generally, when we visited a colony, we would set up our landing station, and we would get ready to survey the colony. For the purposes of UAV surveys, we always use a Phantom 4 Pro, which is a quadcopter rather than a fixed wing aircraft. There's a wide range of UAV types that you can use, but this one is, I feel like, pretty widely available and certainly more affordable than some of the models you see. And then we flew at a variety of heights ranging from 60 meters all the way down to 15 meters, assuming that we didn't have excessive disturbance. And the 15 meters was informed by a study by Rush et al., where they flew about 15 meters above a colony, over a gull colony. The UAV collects a series of overlapping images, and I was able to change the overlap depending on if I wanted to have less of a risk of having missing spaces. And then we also did behavioral observations while the UAV was flying, which I'll get into a little more on the next slide. For colonies where disturbance wasn't necessarily an issue, we did do ground counts of select colonies. And then after we got back from the field, these images were mosaic together. And here you can see an example of one kind of mosaic that we captured. So this essentially is one of our rookery sites. And as you can see, like all of the images were stitched together to just make one cohesive picture that we eventually overlaid a grid on top of. And we will have multiple observers go through and interpret that imagery. And as you can see in this next photo here, this is how we put it in ArcGIS Pro. Yellow lines are a little difficult to see on this picture, but this is how we sort of organize ourselves and facilitate the manual interpretation by individuals. So I mentioned a little bit about our behavioral protocols, and I'll go over it, the general way we go about them. So we have one set of things we do before survey where we record our distance from the colony, and we use rangefinders for this. We also estimate colony size, and we also set up a video camera that we put on the colony, and we try to get as wide a field of view as possible. And as soon as the UAV takes off in the air, we begin counting the number of birds in the air at 1 minute intervals. We also record any behavioral changes that are obvious to us. So if, for instance, a bird is on a nest, and the most obvious would be a bird leaving the nest, we record that and the number of birds that leave. But we also try to record changes in vocalization and other minor behaviors. As the survey is happening, we are also recording the colony's behavior via video camera. Once the UAV returns to its home location, we continue to observe the colony after flights, and if birds have left their nest during the survey, we quantify the number of time that it takes to return to the nest. So now that I've gone over some of our behavioral protocols, we also wondered whether or not we could use UAVs to determine productivity estimates for colonies where we don't necessarily walk through the nest and check each nest for eggs, in particular looking at great blue herons. So with abundance, you imagine going to a UAV colony and capturing a set of overlapping images, mosaicing them together, and then interpreting the entire colony, and you have that snapshot of how many birds are nesting in the colony at one time. For productivity estimates, essentially, it's a very similar process. We just visit these colonies multiple times throughout the season at consistent intervals, and we're surveying the same area over and over again. And when the imagery is collected, it is geo-referenced. So essentially, if you have a nest that's readily visible in one image, you can come back and once that you mosaic the subsequent visit, it has a very similar coordinate, whether that's in UTMs or WGS. For relevant species, we will try to ground count those, assuming that disturbance isn't excessive. And then once we get back, that imagery is interpreted by multiple observers, and we look for changes in nesting activity. So if we come back and we notice that an adult is no longer incubating a nest from one visit to another, that might indicate some changes in nesting activity that we weren't able to detect otherwise. And here's an example of one of our images from a great blue heron colony we captured this year. In two of the nests, you can sort of see you can see the adults as just this whitish blob sitting in their nest. And Logan touched a little bit on some of the image interpretation. In 2019, the U.S. Fish and Wildlife Service flew over the coastal islands, and the survey area was mosaiced and georeferenced. Each mosaic was interpreted by multiple observers. Essentially what we did was we went grid by grid and marked individual birds and assigned them a nesting status or a non-nesting status, as well as their species. And essentially what we would do for the UAV-based imagery is the same procedure. And then to detect observer errors in interpretation, each independent observer goes through the imagery once, and then we compare variation between those interpretations. So I would like to go into some very preliminary findings with the caveat that we're still working with some of the items that we collected this summer. But with respect to behavioral response, we had very little response to UAV from 30 meters to 60 meters across species. There were essentially no flushings or limited flights of birds leaving the nest that we could attribute to the drone from our observations, although the video might reveal a slightly different story. But we are also hoping to continue to look at how the timing of the breeding season affects the bird response. We had no colony-wide dreads, so never during any of our surveys did we have the birds as a colony leave their nests as a huge disturbance. But that being said, we are trying to find ways to measure disturbance at smaller scales, given that just because a bird doesn't necessarily leave its nest doesn't mean it's not stressed. So we're looking at the potential of utilizing things like sound recordings to capture some of the minor details that we can't necessarily see when we're standing 100 meters back or more from the colony. So for estimating productivity, some initial findings, this is very much a work in progress, but we are optimistic because in this picture here, we do have some imagery where we have in the leftmost nest 3 great blue heron nestlings, and you can sort of see the size difference between them and some of the adults that are standing right next to it. And so we're focusing on getting out to sites throughout the entire season next year so that we can readily monitor them and continue to implement some of this work. Some of the caveats for this work is that we do feel like it's very much species specific. So with herons in particular and cormorants that might be more restricted to their nest, this might lend itself better to this sort of monitoring versus gulls, which will leave their nest fairly early on after hatching. And then we are continuing to build upon some of this. So in addition to refining behavioral observations and survey protocols, we are hoping to survey more islands next year to capture some more species diversity, topographic variation, and variation in veg cover. And we are also hoping to get, as Logan mentioned, some pilot surveys with a thermal camera. And so we did bring out a thermal camera with us to a couple of the islands. We didn't attach it to a UAV or anything, but we do have some heat signatures that we're hoping that we can see even from the air. And then, of course, we're developing more protocols for the continued use of UAVs to survey colonial water birds in the Gulf of Maine. And we are continuing to build upon the automated process as well to facilitate the interpretation of this imagery. So this project would not have been possible with without the help of all of our partners, especially this field season. So a really huge thank you to Inland Fisheries and Wildlife and U.S. Fish and Wildlife Service who helped rally to get us some of the imagery data, as well as helping us out to field sites and helping us select field sites. And of course, I had the privilege of working with 2 wonderful undergraduate technicians who were especially flexible given the situation over the summer. And of course, continued appreciation to all of the landowners who let me poke around all of their land to look for birds. And thank you. And you can reach me at this email if you have any questions that are pending after this talk.
Great, thank you, Meredith. We do have time we have about 3 minutes for questions, and there's one in the chat right now. How far offshore have you flown UAVs? And do you only fly from a land base or do you fly from boats?
It kind of depends on your comfort. So we try not to fly from a boat, and part of the reason for that is because, especially on the ocean, and I feel like it's definitely a site-specific thing. When you have currents going and the boat is shifting while you're sitting there, if you're just doing an automated flight, the drone has a GPS error of about 1 meter for the model we run, and so it essentially takes a record of what its home location is, and we'll come back to that location unless you take control of it. So it kind of depends on how much your comfort level is with flying the thing manually. But we generally, if we can get on land, it makes it so that it's a lot safer for the equipment if that's your one of your preoccupations. We have flown from across like islands, or we have tried to fly that are from sites where they're on one part of a site and then we have to cross water, and so we'll have a launch pad that's on an island. It does get harder once you get a certain number of meters away. I'm not necessarily sure of what that upper limit is, but eventually you will lose signal too, depending on the model that you have.
Anyone else? Couple comments just saying great talk and the way of the future. I'm excited to find out, just some of the guidelines. I've had people ask me, just in wanting to fly their own personal drone over a heron colony, say, and I say, hold on, we need to think about how high you need to go, or so this will be really useful to kind of direct people so that they're not disturbing birds. All right. Well, I don't see any other questions. It's just about 2:40, so I'm going to go on to the next speaker. Oh, one more from Dave. Would the thermal imaging allow you to differentiate some species from each other?
I'm not 100% sure if they would give off that different of a heat signature. Our idea with eiders is that they're already very cryptic, but they're very well insulated and their nests give a heat signature. So maybe at certain times of day, they might be more visible. But we are hoping to test on a couple of different species just to see how much that is. The model that we have is pretty sensitive, but I'm sure that depending on the size of the bird, you might be able to get a guess, but I can't necessarily speak to if you had similarly sized birds that might be the same shape, and if they're the same body temperature, you might have a couple of issues, I suspect, with truly knowing which species they are.
All right, thank you, Meredith. Okay, we'll move on to our last but not least speaker, Kayla Smith.
Hello.
Hello.
I'm going to try to start my video and hopefully it's not too glitchy. All right. Hi, everybody. My name is Kayla Smith, and I'm a wildlife biologist with the nonprofit Biodiversity Works. So we do wildlife monitoring and mentoring on the island of Martha's Vineyard in Massachusetts. A lot of our work in the spring and summer focuses on protecting beach nesting birds. So I'm going to be talking about a project we started a few years ago with the trustees of reservations and Massachusetts Division of Fisheries and Wildlife, monitoring and banding black skimmers. So black skimmers are a colonial nesting seabird. They have a very distinctive bill. The upper mandible is shorter than the lower mandible, and they get their name from the graceful way that they skim the surface of the water, searching for small fish and invertebrates. So their odd-looking bill and their barking-like call make them very fun to watch and monitor. In the United States, they nest on sandy beaches. In Southern California, there's a small population, and also along the Gulf Coast and Atlantic Coast. And the colonies can last, or they range from a few pairs to hundreds of pairs nesting. So on the Atlantic Coast, Massachusetts is the northernmost extent of their nesting range, and since 1985, they have been reported nesting at 6 different locations in Plymouth Beach, Plymouth, and on Cape Cod. And the numbers have always been very small, usually between 0 to 8 pairs. In 2012, there were 3 pairs of skimmers that nested on the island of Martha's Vineyard for the first time. and those were the only nesting pairs in the state that year. So since 2012, skimmers have returned annually to nest on Martha's Vineyard, and the island has hosted the majority of the nesting pairs in the state. These colonies usually have fairly good productivity, and most pairs are fledging at least one chick. So since 2017, the number of nesting pairs have increased, and it looks like a nesting colony has become pretty well established on the vineyard. So in 2017, Mass Wildlife, Biodiversity Works, and the trustees collaborated on a black skimmer banding project to better understand the site fidelity, the chick survival, and population dynamics during this expansion at the northern extent of their range. And we've joined several other states along the Atlantic and Gulf Coast that have also started banding their colonies as well. Each state has their own color band that they use. Massachusetts has claimed the color pumpkin, which is a nice bright orange color. So each season, we monitor the number of nesting pairs. We get the dates that nests are laid and the dates that nests hatch. And we wait until the majority of the chicks in the colony are over 2 weeks old. We enter the colony and hand capture as many chicks as possible. And the chicks are placed inside a shaded, well-ventilated, soft-sided clothes hamper as we process them. So we set up a small banding station that's a short distance from the colony, and we take measurements and place a field readable band on each chick, and then return them back to the colony. At this point, we have only banded chicks, but we also hope to one day band adults as well. The 2020 nesting season was an exciting one for our project because we monitored a record number of 15 pairs and documented that 4 skimmers banded during the first year of the project in 2017 returned back to Martha's Vineyard to nest, and these banded birds successfully fledged chicks. We also noticed that 6 of our banded birds from the 2018 cohort arrived mid-season. Now, none of them nested, but they were often seen loafing on the shoreline near the core nesting area. And these birds were most likely scoping out the area, and we're hopeful that they'll return to nest next year. So we banded a total of 36 skimmer chicks since 2017. And that's 44% of the birds have been resighted or reported at least once outside of Massachusetts, either during migration or at wintering sites. So considering the small sample size, I'd say the results are pretty good. And I'm hopeful that the number will go up because we are just starting to receive in reports of our juvenile birds that were banded in 2020. So this is showing a chart of resite observations broken down by state since 2017. And Florida has the most by far, and most likely just because that's where most of our birds are overwintering. And there's also a lot of dedicated birders out there looking for banded birds. But there's a pretty good number that have been seen in New York and New Jersey as well. So here's a map of Martha's Vineyard, the nesting location, and some resite locations along the migration routes. So here's Long Island, Coney Island. There were a couple in Maryland, it looks like. And then for our wintering reef sites, there's been at least 5 birds that have been seen in the wintering areas, all in Florida right now. And there's one site in particular that stands out in Northeast Florida at the mouth of the St. Johns River. It's a popular site, and we've had at least those 5 birds have been seen in large flocks of unbanded and banded birds from other states as well. So this banding project is ongoing, and it's exciting to start collecting more resite data and begin to understand more about our northernmost colony of black skimmers. So I'd like to thank our partners, Mass Wildlife, the trustees of reservations, and also Sheriff's Meadow for allowing us to band skimmers at their Little Beach property. And we're also grateful to all of those that took the time to send us resite information on our banded skimmers. So thank you so much, and I'll take any questions that anybody has.
Thank you, Kayla. See if anybody has anything in the chat. Will you be continuing this work?
Yes, we plan to continue doing it as long as there's funding and time.
I know sometimes we get black skimmer sightings in Maine. I didn't see any of your birds go further north.
No, we haven't had any resites from New Hampshire or Maine yet, but maybe one day.
Kayla, I have a question as to, do you have enough resites of the 2nd-year birds or 3rd-year birds to get a sense for where most of them are spending their time if they're not going back to Massachusetts?
I'd say we don't have enough of that information yet, especially with the 3rd-year birds. We're hoping as we have more birds that are banded, we're hoping that we'll get more and more information.
Okay, we do have a couple questions here. Did you have much gull predation when you were catching chicks? And that is when the adults would fly off.
No, we didn't have any instances of predation. Luckily, that hasn't happened to us.
And then another question is from Pam Denman. After you ban the chicks, do you release them at the edge of the colony or try to release them where you captured them?
We try to release them where we captured them. So usually that's within the area of the colony. They don't usually make it too far.
We've got plenty of time for more questions. Erica has a question. You probably said this, but where do people report sightings?
We would like people to report them to the bird banding lab or Mass Wildlife, Carolyn Mostello. A lot of times we'll find things on Facebook where people have been like, oh, look, I saw a banded bird. And then it'll trickle its way over to us. But yeah, the bird banding lab or Mass Wildlife.
Susan asks, are there other species nesting nearby? In New York City, our skimmers nest near piping plovers and least and common terns.
Yeah, so in 2019, we were only able to band 3 of our skimmer chicks because they nest right next to a decent sized colony of common terns and least terns and piping plovers, and there were also oyster catchers nearby too. So there was a lot going on with the timing where we have to make sure that we're not disturbing where we're disturbing the least amount of birds as possible. So luckily the timing worked out this year where we were able to go in after most of the other species were almost fledged.
Yeah, and Susan kind of echoes that by saying, luckily they are nesting when others are fledging.
Right.
Less the plovers are late.